All right. The haiku. Sing you of sinew, when you sing a new tune of when you grew anew. A little bit of alliteration there, east meets west. Okay. So I had, in the beginning, one of the things that has really gone by the wayside in a lot of these lectures has been the history. Normally I start off with like 10 minutes or something like that of history or whatever. And not because I test you on it, because I don't, but it's interesting. I have, back in my mind, this desire to teach the class entirely from a historical perspective, but that's some other day. But I kept it here. I had mentioned Avicenna in that first lecture, and I'll put a little bit of him in this lecture, because he was the first person to really start thinking deeply about muscles, how they function, and thinking about the anatomy of where there were the origin and insertion and the actions that various muscles had. So here's just one statement that he made. The power of locomotion, or motion, is that which contracts and relaxes the muscles, whereby the members and joints are moved, extended, or flexed. This power reaches the limbs by way of the nerves. And there are as many forms of power as there are movement. Each muscle has its own peculiar purpose and obeys the degree of composite sense. So he's saying a lot right here. This is sort of a summary of many aspects of his thought. Probably the most important aspect of this is simply that he understands that there's some kind of deep connection between the nervous system and the muscular system, right? That like the nerves are what are bringing the power. That's the way he describes it here, to the muscles. All right, and there's that's a very astute observation for the 11th century, where he recognizes that muscle and nerve fiber are special, have a special relationship, and they are different than the other types of tissue in the body. All right, we've already established that. That's kind of been what the theme has been this week. So he has these concepts of composite sense, which are sort of anticipating the notions that Louis Sullivan had about form following function. All right, so Avicenna. And then again, I'd already mentioned Vesalius. It was really this guy that, when we talk about the anatomy that he described, it was really musculoskeletal anatomy. This guy did a remarkable job of showing the anatomical relationships and the clear delineation of the various muscles and their origins and insertions across the body. All right, so what is it all about, this relationship between nerves and muscles? It all starts right here. This is where the story is going to begin for the day, at the NMJ, Neuromuscular Junction, where the nerve meets the muscle. All right? So yesterday I spent a bit of time talking about how excitable tissue establishes and maintains a transmembrane potential, and then uses that in various forms to have some sort of output, and that output being an action potential is where we ended the class. And so we're going to pick up from that action potential at the end of the synapse and the beginning of the muscle fiber, the beginning of the effector. So what is muscle tissue? So I had said on the second day that it's one of the 4 primary tissue types, one of the 2 excitable tissues. And there are 3 categories of muscle tissue. There is skeletal muscle, there is cardiac muscle, and there is smooth muscle. And they each have some unique characteristics. So cardiac and skeletal muscle are both striated, and that's due to the structure of the sarcomeres, which we're going to go through in detail today. And then smooth muscle and cardiac muscle only have one nucleus. And cardiac muscle and smooth muscle are both involuntary. So that means that skeletal muscle is going to be striated, have many nuclei, which in lab the other day I asked you to ponder why that might be within the light of this concept of form and function. That's the structure it has. If it has many nuclei, why? Why is it like that? We're going to explore that today. And then it's striated, all right, which has functional significance in terms of how the muscle fiber works. All right, ooh, I'm warm. All right, so when I talked about tissues and organs, I said there were multiple types of tissues in an organ. So a muscle is an organ, and there are multiple tissues that make it up. Connective tissue, so muscle tissue, of course, nerve tissue we've established, but there's connective tissue. There's even endothelium in the lining of the blood vessels that run through the muscles. So the connective tissue that is in muscle has really 3 layers. And we've talked about one of them in lab yesterday. That was the endomysium. That was the sleeve that goes around each individual muscle fiber. But let's go on the other end of that scale now and talk about epimysium, which is the connective tissue that surrounds the entire muscle. It's the connective tissue that surrounds an entire muscle. Epi, meaning on the outside, or surrounding, mysium, meaning muscle. The connective tissue that surrounds an individual muscle. And this is the kind of connective tissue that it's got a lot of collagen in it, so it's quite, it has some strength to it, we'll say. And it's connected to the deep fascia planes in the body. So there are planes of connective tissue that run throughout the body. Sadly, we won't get a chance to talk about them at all. But the epimysium is connected to these deep planes of connective tissue that run through the body. And the job of the epimysium is to really define the boundaries of that muscle, make the muscle stand out as an individual entity. Yeah. The next step, if you were to look at a cross-section of a muscle, you'll notice that a muscle itself is, if you look at it, there are little bundles within a single whole muscle, and those bundles are called fascicles. Fascicles. So paramecium is a qualitatively different type of connective tissue than epimysium that's going to surround each of these fascicles. And when I say qualitatively different, it's qualitatively different because it acts at the construction of that connective tissue acts as a really excellent conduit for the neurovascular bundles that are going to be running through the muscle. All right, so it's kind of like the service roads for the muscle. You'll have a lot of arterioles and venules and the small nerve branches that are going into a muscle to serve to actually connect with each of the muscle fibers. They're able to pass through this paramecium quite well. And they surround the muscle fascicles and give them some sort of distinction or definition. When a muscle works, when you contract a muscle does not, when you go to move a muscle, the whole muscle isn't, all the muscle fibers aren't firing at once across the whole body of the muscle to cause an action. Sometimes There may be muscle fascicles at this end of a muscle. So, for example, a classic one is the deltoid, right? So deltoid is a three-headed muscle, really, a tripennate muscle. There's a middle branch, an anterior branch, and a posterior branch that get engaged differentially depending upon where in the range of motion you're trying to move that joint. And this is the job of the various fascicles, right? And they're going to have individual nerve branches that are controlling them, etc. The next layer we've already talked about in lab, and that's the endomysium. Endo meaning on the inside, the very innermost layer of connective tissue. So if you were to pull one of these fascicles out, it itself would be a collection or a bundle of little muscle fibers, muscle cells. And each of those cells is going to be surrounded by a sleeve of this endomysium. So the very smallest blood vessels, the capillaries, are going to run through the endomysium. And this is also going to be where we find what are called myosatellite cells. The myosatellite cells are basically muscle stem cells. And they're not called, well, let's just leave it at myosatellite cells is the name that we give them. And their job is, say you tear a muscle or maybe you need to add muscle fiber for some reason, you are going to be mobilizing these myosatellite cells. All right, so let's look a little bit more closely at an actual skeletal muscle fiber. The thing about them is they're remarkable cells. They're huge. They're really big cells compared to a lot of the cells that you could find in your body. And this is because they are formed through the fusion of these mesodermally derived cells called myoblasts, all right, which is sort of a not fully developed muscle cell. So we've encountered that suffix blast before in very similar or analogous contexts. They have hundreds of nuclei because you have these myoblasts fusing together. Each of them is bringing a nucleus with it. Why is that? What's the functional significance of that? What do you guys think? I've asked you this a while ago. Yeah, what do you think, Miles?
Well, nuclei are like starting place for protein formation.
That's right.
And muscles have a massive amount of protein in them.
That's right. That's right. Okay.
And they're also long, so you want to have like an even distribution of protein-making centers.
Thank you. That's exactly it. You guys follow that? So you have these enormous cells. They're enormous because they're doing something mechanical, right? If you have a single cell that is actually quite long, then you don't have to worry about how one cell is connected to another as you're putting tension on it. It's just one continuous, instead of like splicing ropes together, you just have one long, strong rope, okay? And if you're having this long, strong rope, sometimes you're going to be needing to repair it, right? Because you've damaged it somehow or whatever. And just as he said, it helps you put protein where you need it in the cell without having to transport proteins around it would be a logistical problem to get proteins around a large cell like that. All right, so this is kind of a pointless slide, I suppose. I could have deleted, but I guess the only thing new here is to just point out how big some of these are. 30 centimeters. 30 centimeters. That's about a foot long. This is about 30 centimeters right there. That's a large cell. That's a very large cell. Okay. So let's go through the anatomy again, the parts. We talked about it in the lab, but I want to go over it here so we can talk about what each of these parts does. So here's our diagram. We have the sarcolemma surrounding the outside of the cell. All of it is an excitable membrane. It communicates with these T-tubules, which in this diagram are yellow. On the model in the lab, they're blue. The blue stuff here was yellow in the lab. The coloring is unfortunately opposite, but all of it's sort of a pinkish color in the body, I suppose. A muscle cell itself is a bundle of these things called myofibrils. So a myofibril is this itself collection of myofilaments, these thin and thick filaments that are bundled together. So within a single myofibril is the functional unit called a sarcomere, and that's basically one set of stripes in a striation, a pattern of striation. This blue here is called the sarcoplasmic reticulum. To remind you, we talked about it. It's derived from endoplasmic reticulum. This ends up being a place where calcium is stored, and we'll see how that plays into the whole mechanism in a moment. These triads are a collection of transverse tubules, which are, remember, just little invaginations of the sarcoplasmic sarcolemma. Pardon me. So this is all going to be excitable membrane here, and it's directly adjacent to these terminal cisterna, which are these cisterns or these wells of sarcoplasmic reticulum, and all of it is bundling up one of these myofibrils. Is that everything? I think so. Oh, yeah. So in these cells, one other thing is there is a red pigment called myoglobin, and it is related to hemoglobin, except hemoglobin, I don't know, has anyone had any biochemistry here? No? You guys are all like sophomores and juniors. Biochemistry, in a biochem class, they would talk about hemoglobin, where this is a protein that has 4 subunits, right? A tertiary structure, 4 subunits. It's an oxygen-carrying protein, of course, in our blood. Myoglobin is a single subunit, all right? So it doesn't have the same what's called allosteric properties as hemoglobin has, but it is an oxygen carrier, and so it serves as a reserve of oxygen in these cells, and it's one of the reasons that muscle is particularly red. Muscle tissue is particularly red. Okay, so let's look a little bit closer at the sarcomere. I've got a little scanning electron micrograph here, and I'm only doing that to help explain A and I. And I guess I should put the spelling of isotropic and anisotropic, but I don't know. If those words don't mean anything to you, maybe just remembering I band and A band is enough. It's only one letter instead of a whole word. But this region of the sarcomere is isotropic. We'll see why when we see the structure of those proteins, but it essentially means that those proteins have a handedness and they spin light in a certain polarized light in a certain directionality, which is something that was discovered when these microscopists were using polarized light on the samples. And then there's the anisotropic region, the A band. It turns out the A band is made up of thick filaments. The I band is only thin filaments. There's this region in the A band called the zone of overlap where thin filaments and thick filaments coexist. This is truly where the rubber meets the road in terms of contraction. There's a little region in the middle called the H band, which is just thick filaments. I don't remember why they call it H band, but it's just where there's no thin filaments. And down the middle of this, there's this M line, and down the middle of the I band, there's the Z line. So M standing for not middle, but Mittel, which is middle in German, and Z for Zusammen, meaning this is where 2 sarcomere are next to one another. Okay? So let's look at cartoon to get a better sense of what this looks like. So the Z line, maybe it'll help you think of it not as Zusammen, for those who aren't into German, maybe just zigzag. This zigzag line that runs down the middle of the I band. And then M line, this is the middle of the sarcomere. Okay. So when we see here in blue, these are the thick filaments. In pink, these are the thin filaments, and we can see them overlapping in this zone of overlap. There's this protein called titin, which connects the end of a thick filament to the Z line, and it kind of does a couple things. It helps the filaments not turn into a bowl of spaghetti. It helps everything stay lined up properly, and it also helps in terms of recoil. Stabilizes the filaments. All right, so let's just look at some cross sections so we can kind of get a three-dimensional sense of this maybe. Right at the Z line, there's this beautiful sort of hexagonal mesh of protein where we have titin, but the titin that anchors the ends of these thick filaments is bound in this meshwork to each of the thin filaments, the origin of the thin filaments. And then cut through here, we just have thin filaments with titin intercalated. In the zone of overlap, we don't actually have a slice through there. Oh, I guess we have one here, yeah. So at the M line, we just have the origin of all the thick filaments. This is somewhere through the H band. And then in the zone of overlap, we have these thick filaments surrounded by these collars of thin filaments. And it's going to be this interaction between what's going to turn out to be actin, which is the thin filament, we'll see that in detail in a minute, is going to interact with myosin, which are these thick filaments. Okay, so let's get some of the characters fleshed out a little bit, no pun intended. First is F-actin. F-actin, filamentous actin, is what we call the thin filaments. So F-actin is a helical, a double helical twisted chain of protein, like a protein complex, right? And so running down the middle is this protein called nebulin. It just acts as the thread down the middle. And then twisting around it is these two spirals of these sort of beads on a chain. And each of these beads, we're calling them beads, although the actual protein has some obviously stranger shape. This is G-actin. G-actin is globular actin. It's a globular protein. So there are filamentous proteins, there are globular proteins, there are transmembrane proteins. There is this chain of globular proteins, right? And on those globular proteins, on the G-actin, there are active sites. There are active sites. This is the part of the protein that's going to actually interact with myosin, the other protein, the actual motor protein. G-actin is not a motor protein. It really is just sort of the track. It's the track that this motor protein is going to run on. And the part that motor interacts with is depicted in yellow here, the active site. Now, that active site, when a muscle is not being used, is covered up. It's covered up so that myosin cannot interact with it. It's being occluded by this protein complex that's made of troponin and tropomyosin. So really it's being covered by tropomyosin, which is this blue twisted protein that we have here. It's this double-stranded protein that lays right down on top of the active site. And then there's this protein called troponin, which appears every 8th G-actin. Every eighth G-actin, you're going to have a troponin that is going to sit right on top of that complex. And it is troponin which actually holds the tropomyosin in place. It's what holds that tropomyosin in place. And the troponin is an extremely important protein because this is the protein that is going to act as the trigger. It's the regulating, it's the regulatory protein that is going to roll the tropomyosin off of the G-actin or keep it on, depending upon the molecular signal that's in the cell. Does anyone want to guess what that molecular signal is? Go back, Thank you. You win another invisible cookie. That's right. Yeah, calcium. Oh, it says it right there, isn't it? Right, so troponin is going to bind calcium, and when it does, it's going to roll tropomyosin off of the active site, and we're going to have a contraction, all right? So we're setting it up here, setting it up. Let's switch over to the thick filaments. So the thick filaments are these bundles of myosin subunits. So here it is. It kind of looks like a golf putter. It looks like a putter, right? But it's like a Jetsons putter, some like futuristic putter where that you don't even have to swing it. The like head does it itself. The head has got a little hinge on it, and it stores energy in that hinge. And it's the motion of this hinge which is actually what is going to help to cause a muscle contraction. So a number of these putters get all bundled up together into this long, thick filament. All right. So when I talk about isotropic and anisotropic, if you look at this, there is a handedness to this spiral. All right, so this is going to spin polarized light in one direction. All right. Whereas this, although the artist sort of drew this as a spiral, this is anisotropic. This is not going to twist light. It's just sort of a bundle of these things that runs along the shaft of the thick filament. All right, so light is randomly dispersed as it passes over that. Polarized light is randomly dispersed. If that is confusing to you, you can strike it from your mind. It's okay. Okay, so this is not the whole cast of characters. This isn't the whole cast of characters, but this is everything that we're going to need to talk about the very basics of the mechanics of muscle contraction. But I'll show you a couple others here because there's some interesting pathology that's associated with some of the other players. So it's fine to get these muscle fibers to be contracting with respect to one another, but you have to transfer that motion through the muscle cell, then through the connective tissue and into the bone to make the bones move, right? So there's a lot of links in the chain. Some of the links that just get the contraction at the myofibrils to the surface of the muscle fiber are these four. We have various linking proteins, et cetera, et cetera, et cetera. The one that's important to us is this one, and it's called dystrophin. We call it dystrophin because it was identified as being, the gene that encodes dystrophin was identified as being the causative agent for muscular dystrophy. All right, and muscular dystrophy, dystrophy, is simply a word that means improperly formed, something that doesn't take the proper shape or form. And so in muscular dystrophy, the muscles are dystrophic. They don't form properly for some reason. They don't have the right shape. And it's because this protein doesn't work. This protein doesn't work right. And so the contractions that happen in the muscle fiber are not able to be transmitted to the surface of the cell and contract that cell, and then translate that further into motion, etc. So, okay. Again, this slide is purely a part of the weirdness of my mind that likes to look at broader patterns in nature, the underlying physical forces that guide biological structures. And so I'm not going to delve deeply here, but I'm just going to like open the window a little bit for those who like to peer out onto beautiful landscapes, and point out that muscle tissue is fractal in structure. So what a fractal? We've probably all heard that word. Maybe most of you know it, what it means, maybe not. A fractal is something that means there is this scale invariant self-similarity. So there's like some sort of pattern that gets repeated in some system across many size scales, all right? Like a snowflake is a fractal structure, and muscle fiber certainly is that. So if you look at it, we have skeletal muscles, which are these bundles of muscle fascicles that are themselves surrounded by a sleeve of epimysium. A muscle fascicle is a bundle of muscle fibers that is surrounded by a sleeve of paramysium. A muscle fiber is a bundle of myofibrils that is surrounded by a sleeve of endomysium. A myofibril is a bundle of myofilaments that's surrounded by a sleeve of sarcoplasmic reticulum. And these filaments, these myofibrils here, these filaments, is simply made up of myofilaments that themselves are compositions of twisted proteins. So I don't know, it's kind of a beautiful structure. Okay, so my goal for the day is to talk about the mechanism of contraction on one hand. And then once I do that, time allowing, I would like to talk a little bit about how we pay for it, right? It's one thing to like build a wall, it's another thing to pay for it. And that's what we're going to do. We're going to talk about actual contraction and then how it gets metabolically paid for, all right? And so within that, with that in mind, we've established the protein elements for the contractile aspect. We have all the pieces in place, but I wanted to show this picture here because one of the things that this picture shows really well, we can see the sarcomeres here. Here's the Z line. Here's the I band. Here's a big A band, the M line. This would probably be some zone of overlap or whatever. But then there's all this other smudge in between. Okay, all this stuff here. And this stuff is mitochondria, which are the powerhouses of the cell. They take, what do mitochondria do? They are responsible for taking molecular breakdown products of glucose, specifically pyruvate or acetyl-CoA, and they take that molecule and they turn it into ATP. But to get the pyruvate and acetyl-CoA, we need sugar. We need sugar, and so that's what all this stuff is here. All this brown stuff is glycogen. Glycogen is simply animal starch. That's all it is. Has anyone not heard of glycogen before? Okay. Well, anyways, that's glycogen, and we're going to see how all that works at the end, time allowing. So the sliding filament theory, is this? Yeah, that's the slide. Okay, So the way actual contraction works, before I talk about the molecular details of the mechanism, I just want to get the broad mechanics of it in your head. So what I would like is some odd number of students to come up. Maybe like 5 or 4. I can be one of them. Come on up. Who wants to move around? There's a chance to get out of your seats. Yep, that's right. Perfect. Wonderful. Okay, so I'm going to have you two be, what are you going to be? You guys are going to be thick filaments, and we'll be some thin filaments, the 3 of us, okay? So no, you're actually right. We'll put him in the middle, and you're right there. Okay, let's spread it out. Put your arms out like this. Okay, and so now, the way this is going to work is, you guys are thick filaments. So what you're going to do is you're going to, and we're just going to stand there and not do much. And they are going to grab a hold of you, and they're going to go like this. Go, whoa. Okay, they're going to grab hold, and then they're going to do that with your wrist and sort of pull the person closer. All right, pretty simple demonstration. So you do it, and ooh, wow, yeah, go for it. We're contracting. We're contracting. Okay, you guys get it? So you feel that, right? Did you feel it? You're doing it, like grabbing them, pulling them? Okay, good. Did that illustrate something? It's this idea here. Oops. It's that the thin filaments and the thick filaments aren't getting shorter or longer. Oops. They're just sliding over one another. That's all. They're just sliding over one another. Like our arms didn't get shorter. They just overlapped more. So what's really happening, if you look at this, is the zone of overlap is changing in its size, all right, in the relative proportion of the sarcomere. That zone of overlap is changing in its relative proportion. So zone of overlap goes up, I band and H band go down. That's all. But the width of the A band stays the same. Okay. So that's the broad mechanics of it. And this is an overview of the molecular details. So the first thing that happens is we decide to move a muscle. So I'm standing here just minding my own business, and then a werewolf, a politician werewolf, comes running in to the room here, and I got to move, I'm going to run. So my brain sends a signal to my muscles to move, right? And that signal comes down through the nerves, and that nerve releases acetylcholine onto the surface of the muscle. So we get this excitation happening. There's going to be an excitation that happens along the surface of the membrane. Then there is this set of events that couples excitation with the actual contraction that's happening. That's called excitation-contraction coupling. So all of this calcium release, this actin-myosin interaction, this is going to be the excitation-contraction coupling. Then there's going to be actual muscle fiber contraction, which was going to give rise to the buildup of tension, and that gives us excitable membranes. Well, all right. There we go. One other point that I wanted to make is that the tendons that you see in these muscles here, the tendons are not, it's not like some new thing that gets incorporated into the muscle at the end. It's really just the epimysium the paramecium and the endomecium of the muscle coming together and the muscle fibers themselves running out. So it's just a continuation of that connective tissue, those connective tissue sheaths that sort of come together in a rope at the end. That's what a tendon actually is. Okay, so here are those 4 phases. There's excitation, and this is where the nerve is going to send the signal via action potential and cause a depolarization across the membrane of the sarcolemma, across that sarcolemma. And action potential is going to happen in the muscle cell. Then there's this excitation-contraction coupling. This is a sequence of events that is going to link the action potential at the neuromuscular junction, at that motor end plate, to activation of the myofilaments inside the muscle cell. And then there is going to be a sequence of contraction. So this like cycle, this contraction cycle that will happen and happen and happen until the muscle fiber relaxes. And then finally there's relaxation, and that's how we reset the situation. All right, so we'll go through them with little pictures here, one at a time. On the right, we see a picture of muscle fibers with the telodendria of a neuron ending at this motor end plate on each of the muscle fibers. Here it is as a cartoon. So here is the synaptic bouton or synaptic knob, and we have the little vesicles that are filled with acetylcholine. Acetylcholine is the neurotransmitter or the chemical that is going to activate these chemically gated channels, ion channels, over here on the other side. So, yeah, we're ready to go. Let's do it. Signal comes down. Signal comes down along the axon to the end of the synaptic knob. That depolarization is going to enable these vesicles of acetylcholine to fuse with the membrane here and release their acetylcholine into the synaptic cleft. All right? Once that happens, acetylcholine binds to the acetylcholine receptors, opens them up to the influx of sodium. So remember what the membrane dynamic that we had talked about previously, where you have a lot of sodium outside the cell, a lot of potassium inside the cell, about a minus 70 millivolt resting potential. Okay? And that sodium electrochemical gradient wants to come inside. The potassium electrochemical gradient wants it to go outside. So acetylcholine receptors open up and let that sodium blow into the cell. And it starts a broad depolarization on the membrane of the muscle cell. Now, when this happens, this is excitable membrane. This is excitable membrane, meaning not only does it have chemically gated ion channels, it also has voltage-gated ion channels. All right? It also has voltage-gated ion channels. So that when we're tripping it right here, we're triggering it with the acetylcholine, but there's other channels here that are going to get set off. Okay, so I don't know. This slide is kind of silly, but it's just a picture showing those acetylcholine receptors. Let's go here. So here are these voltage-regulated ion channels, and it's showing them open at the same time. That's not actually the way that works, but you can see that it's right next to these acetylcholine receptors. So this is the cast of characters that we have, and in fact, it's actually missing a couple other characters. Can you guess what those characters might be? What else do you think we need here? Huh? We need some leak channels. Sure, yeah, there's definitely going to be some leak channels. But if there's leak channels, what else is there? Pumps. The sodium-potassium ATPase, that antiporter, right? So those are two other things that are going to be in here to make this membrane work the way it's supposed to work. Everybody follow that logic? If not, raise your hand. That's why I'm here. I get paid for that. And so it be, no, I'm not complaining on camera. Okay. Yeah, so action potential gets tripped and it starts rolling across the surface of the membrane. Meanwhile, back here at the motor end plate in the synaptic cleft, the acetylcholine is in equilibrium with those receptors, right? Like it binds and then it comes off and then it binds again. That's just how molecules work, right? There's these equilibrium of association constants. And when they come off, these half-moon proteins here are meant to represent something called acetylcholine esterase. Acetylcholine esterase is an enzyme, so any kind of enzyme that you hear, any kind of protein that has ACE as the end is going to be some kind of enzyme, which means it's going to catalyze or facilitate a chemical reaction. It's going to make a chemical reaction go more quickly than it normally would. That chemical reaction that these are doing, it's an esterase. That means it's going to cleave or break an ester bond, an ester linkage. So there's an ester linkage that connects an acetyl group to choline. So acetylcholine esterase breaks acetylcholine into acetate and choline, making it not usable any longer on these receptors, freeing it for recycling. So this choline that gets released gets taken up by the cell once again, the synaptic knob. There are choline receptors, choline transporters that suck the choline back up, and when the choline gets taken into the neuron, it gets quickly acetylated again. It has an acetyl group stuck back on it, and it's ready to go once again. Okay, so pretty nice little molecular machinery there for turning that trigger over. All right, so that's an excitation. We need to couple excitation at the motor end plate with the contraction, which is what we actually want. All right, so excitation-contraction coupling. This action potential is rolling across the membrane, just like we saw it yesterday. It's rolling across that membrane. All those voltage-gated ion channels are swinging open. Sodium's pumping in. Comes down, and it gets to these T-tubules, and it enters the muscle fiber at this triad. So remember, this is the terminal cisterna of the sarcoplasmic reticulum. This is the T-tubule. This is another terminal cisterna. So we have depolarization happening here. The depolarization that happens in this T-tubule triggers an event here in the endoplasmic reticulum that opens up its own ion channels, calcium ion channels. All right. So calcium that has been sequestered in this sarcoplasmic reticulum is going to roll down its electrochemical gradient. It wants to get out of here, where there's a ton of calcium and no anions. It wants to get out and be near all the anionic charge that's inside the cell. It's going to go down its electrochemical gradient. So that's it. That's the excitation-contraction coupling. This signal comes in and triggers the release of calcium from the sarcoplasmic reticulum. So there are like pieces here that I'm not filling in for you, right? There's got to be calcium pumps, right, in the sarcoplasmic reticulum for like chucking the calcium back into the cell there. And if there's a pump chucking it in, there's got to be some leak channels, et cetera, et cetera. And there's proteins that mediate this interaction. Okay, so here we are before excitation. We have the thick filaments with the myosin here, the little head groups, the putters, the Jetsons putters. We have the thin filaments with the G-actin active sites covered by the troponin and tropomyosin, the troponin that's holding the tropomyosin in place. But now calcium gets released the way I just said it did. Calcium comes out, binds to troponin, troponin rolls tropomyosin out of the way, and now we can form a cross bridge between the myosin head group and the active site on G-actin. All right. Now we're ready for the contraction cycle. So at the beginning, when we're starting at the beginning, we kind of already put one in the bank, so to speak. We had one in the bank from last time. So here's our myosin. That head group is open, cocked, and there's a bunch of energy that's stored in this hinge, potential energy. That energy came from the hydrolysis of ATP into ADP and inorganic phosphate. And it's just sitting there, still bound. It hasn't been released. It's still bound to it, but the energy of that bond has been stored in this hinge region. Calcium comes. We see the active site revealed. We form. As soon as we reveal that active site, we're able to form a cross bridge. Now, should I go here? Maybe I will. It's kind of interesting. I think it's really interesting. But so the way molecules work, so, you know, it's a different world down there in solution. It's a different world in solution. And maybe this isn't for everybody here, but I'm going to give it to the few of you who want to think a little bit more deeply about it. Like the biophysics, because that's what I am. I'm sort of interested by the biophysics of this. When two things interact with one another, they have to get through water. And water to us, we dive in, we swim through, no problem. But if you're a teeny tiny little molecule, water is really hard to get through sometimes, right? It's like hydrogen bonded to itself and like to everything. It's kind of viscous, a lot of surface tension. It's kind of hard for stuff to make it through sometimes. And so cells have taken, biology has taken advantage of that. When something is in solution, it orders the water that's around it, right? Because the water, in bulk, water can form all kinds of hydrogen bonds any which way it wants. If you look at a picture of water, bulk water, it's got all kinds of crazy hydrogen bonding patterns that are constantly shifting. Super chaotic, right? Entropy, a lot of entropic component to that. But when you stick something in there, a solute, a protein, that doesn't have nearly as many possibilities for it to hydrogen bond and not as many states that water can take, the water becomes slightly ordered around that solute. It becomes ordered around that solute. And that ordering is not just in the first layer. It extends out into solvation spheres. All right? The specific way, because, the water that's really ordered, the guys that are next door to it, they get ordered too. I don't know why everything is relating in my mind to the government shutdown, but it's like these government workers aren't getting paid, but then the people with the food stands, the like hot dog stands on the corner in Washington, D.C., they're in dire economic straits because they can't sell hot dogs to the government workers because they've been furloughed for 2 weeks, right? So there's these layers. There's these layers in the economy. It's not just 800,000 people that are out of work right now. It's a much broader part of the economy. It's the same with this, right? So these solvation spheres are move out. And it's not just we can think of it in the abstract and the general, but there are very unique, it's like a fingerprint. Like those people who don't have jobs and the people who aren't selling enough hot dogs, they're real people, right? They're not just abstract things, they're actual real people. And this shutdown is like other shutdowns in the past, but it's unique because those people were never affected before. It's just those people and their jobs and that life. And it's the same with any of this. Like everything that's in solution has a unique pattern, to how it perturbs the water. So for something like this myosin head group to come and bind, the solvation pattern, the way this active site is going to perturb the water that's sitting right here, is going to perturb it in such a way that this myosin head group, and this is evolution, right? They evolved so that myosin head group, as soon as the water gets perturbed in that way, it's able to work its way through it real easily. There's something about the way this solvation sphere gets perturbed that it becomes really easy to form a cross bridge. This thing doesn't, like the myosin doesn't just, or the actin just doesn't say like whistle and say, hey, I'm ready, let's go. It communicates that at a short, albeit short distance, but at a distance via the water and the solvation sphere, and how that changes when these activation sites get opened up. And you can think about that much more broadly in terms of how any kind of proteins are going to interact with one another. You can imagine like your immune system. I know I'm going on a tangent here, but it is a really fascinating thing if you dive into it. How does one antibody find, like maybe I have only a few antibodies that recognize some antigen and they're in my toe. How are they going to know to get some antigen or pathogen that is in my finger, right? How do all the molecular interactions that need to happen, the specific molecular interactions that need to happen, sort themselves out when if it were purely stochastically random, like everything was just bumping and sampling each other thing that they needed to bump into. Things need to sort at a distance, and it's not like that. Like proteins can text one another and coordinate, right? It's through the solvent medium, and so that's another constraint on the form of something. Another constraint on the three-dimensional form. When you see these proteins that have these complex structures, a lot, and you're like, oh, there's tiny little active site, but then there's this huge protein. Oftentimes, it's about modifying the solvent environment around a protein. And this is what often the glycosyl portion, the carbohydrate portion that gets attached to a glycoprotein. Carbohydrates have all these hydroxyl groups, and it changes. Okay, that's enough of a tangent. I got my heart going there. But maybe it'll help you appreciate how this stuff works. And it's not just magic and cartoons. There's a lot of real biophysics that is underlying the evolution of this. Yeah?
It's just like, to kind of put it in more layman's terms, like it's kind of like this cascade of action is like changing the texture of the water, right?
Sure.
And like that change in texture is causing it to be easier for this whole reaction to happen.
Yeah. Imagine it like this. Say you're at a concert. And you're about, 20 rows back and you want to get up to the front and it's like everybody's milling about there. But then somehow magically they throw one of those giant beach balls on this side and one giant beach ball on that side, and everybody in the crowd who's just milling about, half of them turn this way and half of them turn that way, and then suddenly there's this beautiful path through the crowd and you just zip right through it, right? That's kind of what I'm talking about. It's like a rough analogy. Okay, so let's get to actually making the muscles work here. Cross-bridge formation happens. As soon as that myosin head touches the active site, it's going to cause a change, a slight conformational change in that head, where it's going to kick off the ADP and pi. They're going to not be held anymore. All right? They're going to be released. As soon as they come off, they're like the linchpin in this. As soon as the ADP and inorganic phosphate come off, all that energy that's stored up in the hinge comes out, and we have this little. That's it. That's a contraction. That's a contraction. It moves. It moves this thing, the actin fiber, that way relative to the myosin. As soon as the hinge releases that energy, that change causes another slight conformational shift, which disattaches the head from the active site on actin. Once that happens, if you have a hinge, a head that has been fired off, that's not attached to anything, that is the perfect scenario for ATP to come. ATP to come and bind. ATP is then able to bind. As soon as ATP binds, this head group, amongst its many functions, I've just given you, if you like micro dissect everything that it's doing, all the conformational shifts that have to happen, it has so many functions in this small little head. It then binds the ATP, and it's an ATPase. It hydrolyzes ATP. As it hydrolyzes the ATP, that energy gets stored back in the hinge, and it comes back. Seems like a simple thing, right? It's just doing this. But each of those steps, it has to recognize the actin, bind the actin. When it binds the actin, it goes through a conformational shift and releases the ADP and inorganic phosphate. When that comes off, it causes another shift and that makes it release. And then it's going to bind, there's all of these little steps that are constraining the evolutionary shape, the development of that myosin head group. It's pretty remarkable if you really think about it. Okay, so here's the contraction cycle. And it just, when you go through a contraction cycle, it just shortens the length of the muscle fiber. Oh yeah, look at that. Okay, too much fun with PowerPoint. So how long does this last? Well, it's dependent upon a number of things. So first of all is the duration of the neural stimulus. All right, how rapidly is acetylcholine esterase turning over the acetylcholine, for example? Like how long is the acetylcholine dwelling in that neural cleft, or in the synaptic cleft? Also, how much free calcium is available in the sarcoplasm, because that's really what's doing it, right? If there's calcium there to bind to tropomyosin and troponin and keep the tropomyosin off the active site, then it's going to keep going. But we have to pay for it, right? And we pay for it with ATP. So the contraction duration depends on how readily available ATP is, our energy source. And this was getting at the other thing that I wanted to talk about, how to pay for it all, right? So I'm going to clean up a few details, and then we'll dive into some ATP. First, let's figure out how to get the cell to relax. So here we go with the acetylcholine esterase. It breaks up the acetylcholine into not tiny little bits like that, but an acetate and a choline. Choline gets recycled, reacetylated, and packaged into new vesicles. All right, so that's the first thing that has to happen. We need to do this so that that transmembrane potential can reestablish itself. All right? Once that happens, the calcium pumps, so the calcium channels close, those calcium channels that were triggered by at the T tubules, these calcium channels close in the terminal cisterna, and the pumps, the calcium pumps, take over and gets pumped back into the sarcoplasmic reticulum. Importantly, by active transport. What does that mean? What is active transport again? Yeah.
It's going against its gradient.
It's going against its gradient. What does that require? ATP. Thank you. Yeah, so when you're done contracting a muscle, you're not done burning ATP. All right? That's an important take-home. When you're done contracting muscle, you're not done burning ATP because you got to put all the pieces away. It's one thing to play with your toys and you're exhausted, but leave a little energy to clean up afterwards. Oh, was there another point here? Yeah, okay. No. So we got to get the muscles back to their resting length. And there's a number of things. First of all, there's elastic forces. All right, so the tendons and ligaments that are involved in muscles are going to pull that muscle fiber back to its supposed resting length. There's also the force of opposing muscle contractions. So I can lift this heavy weight here, and I've contracted this muscle, but I want to relax this muscle. I'm going to use the force of my tricep, the opposing muscle on the backside. And as I do that, it's lengthening this muscle back out to its resting length. And then lastly, thankfully, there's gravity. Just let gravity take over sometimes. That's the best thing you can do. How's everyone doing? Everyone need a lap? Should we do a couple laps? Go out in the hall and come back in. Everyone good? You're considering it. All right. So here is the summary. Should I go through a nice summary? All right, I'll do it. Nerve stimulation comes through the axon in the form of an action potential. Causes the fusion of synaptic vesicles with the presynaptic membrane, releasing the acetylcholine, or that neurotransmitter, into the synaptic cleft. That acetylcholine binds to chemically gated sodium channels and causes them to open. Sodium rushes in, sets off a cascade of dominoes that is because of the voltage-gated sodium channels that are also in the membrane. So an action potential begins to spread across the muscle fiber, down into the T tubules, which are right next to the sarcoplasmic reticulum, the terminal cisterna of the sarcoplasmic reticulum, causing that part of the sarcoplasmic reticulum to release calcium into the cytoplasm. That calcium binds to troponin, which is holding tropomyosin onto the active site of G-actin, and rolls tropomyosin off, revealing the active site on actin. Myosin head group can come across and form a cross bridge formation. When the cross bridge forms, it kicks off ATP or ADP and inorganic phosphate, thus releasing the energy that's been stored in the hinge region and causing a contraction. As it slides, as the head group releases that energy, it goes through another shift in conformation that enables it to disattach from the myosin substrate, and then enables it to bind more ATP, which hydrolyzes to ADP and inorganic phosphate, recocking the head, and we go through a contraction cycle until acetylcholine esterase back at the motor end plate in the synaptic cleft takes the acetylcholine and breaks it down so that the acetylcholine receptor is able to close, and we reset the membrane potential, thus stopping excitation, and calcium pumps begin their job of pumping calcium back into the sarcoplasmic reticulum, releasing calcium from troponin. Tropomyosin gets rolled back onto the active site, and the contraction cycle is over. All right, so that's how it works. So it's a lot, I know, but you'll get it. Okay. Rigor mortis. So, rigor mortis is a fixed muscular contraction after death. And what happens here is that the ion pumps cease to function. Ion pumps cease to function because we don't have any more ATP. The cells in our body aren't getting the oxygen we need. The heart's not pumping. There's no oxygen. The mitochondria don't have the oxygen they need to convert acetyl-CoA into the energy of it into ATP. Cell runs out of ATP, and there's now no longer any ATP to pump calcium back into the sarcoplasmic reticulum. So calcium is just awash inside the cytoplasm. So it's bound to troponin and tropomyosin, and that active site is revealed, and that cross bridge forms, but there's no more ATP to facilitate release and resetting, and et cetera, et cetera. So the muscles just get stuck. The active site, the head gets stuck to the myosin, or the myosin gets stuck to the actin, and you can't reset it. So this is called rigor mortis. It eventually releases after a day or two when the muscle just begins to degrade and all that breaks down and begins to release. So it's not forever, of course. Rigor mortis. Okay. So the way it works, I pick up this light thing here, or I pick up a heavy object, and I feel like, oh, I'm really using my muscle here, but this one is so light, I'm just using it a little bit. And you may think, oh, I'm just contracting this muscle fiber a bit, or now I'm going to really contract that muscle fiber. No, that's not how it works. If you're contracting a muscle fiber, it's doing its thing, whatever that is. All right, and your ability to moderate the amount of pressure that you generate in a muscle is simply a function of how many muscle fibers you're recruiting for that job. For a very light task, you're only recruiting a few muscle fibers, and if you're lifting something very heavy, you're recruiting many of them all at once. So this means that just like any action potential, it's all or none. Just like an action potential in a nerve fiber, if you knock over the dominoes, you're riding the train, okay? And it's the same here. If a muscle fiber starts the contraction cycle, it's going to go through its whole contraction cycle. So the tension that a single muscle fiber can create depends on the number of pivoting cross bridges. So this is a function of the resting length. The resting length. So I'm like sort of sneaking my way over to some yoga for you guys here. You'll see. It also depends upon the frequency of stimulation. So how rapidly are you trying to contract that muscle fiber? Are you contracting it before you're letting it completely release to its resting length? Okay, so that way you can step up the pressure. We'll see pictures of all this in a moment. But the number of pivoting cross bridges is dependent upon the fiber's resting length, and we'll see what implications that has in a minute as well. This is that minute. Okay, so for any, particularly athlete, but anyone in here who's trying to like use their body, this is actually maybe one of the more useful slides I'll show you. And that is the relationship between tension and the length of the zone of overlap. All right? The length of the zone of overlap. So there is this zone right in here where you get maximal tension for the amount of, shall we say, rubber on the road. Okay? If you have more, so I said the more pivoting head, the more head groups attached to the actin that you have, the greater the tension, but that's not a linear relationship. You have more here, but the problem is there's nowhere to go. There's nowhere to go. This muscle fiber is already highly contracted, right? It's already highly contracted. There's not much else where the muscle can go, so they can't generate that much more tension because it can only shorten a little bit more, just a little bit more. On the other hand, you're way over here, you can't generate any tension because there's no rubber on the road. There's hardly any, this muscle is too long. So this would be analogous to someone who maybe tore a muscle or an overly lengthened, overly stretched muscle. This would be an example of some muscle-bound person who's been like exercising, working a muscle, building up muscle density, but never lengthening the muscle. And so they actually don't have as much power as you may suspect they do given the muscle girth, the muscle mass that they have. The point that I'm trying to communicate here is that athletes, a lot of athletes live right in here somewhere. A lot of athletes live right in here. You can get more power out of the same muscle mass just by achieving the optimal length of muscle. So in terms of training, this is why stretching is so important. Like the right kind of supported stretching regime, it will actually increase your power output, the efficiency of contraction in your muscles without having to add any extra mass to your body. Is that like a message that's ringing? Okay. Yeah?
You know, stretching after performance is less effective than stretching while your muscles are relaxed.
Well, so it depends on a couple things. First of all, the specifics of your muscle tonus, the like fundamental muscle tonus that you have to begin with. So making a blanket statement is a little bit hard. Where do you exist anyways? Like, where are you on this anyways? There is benefit to, so stretching, I think, is important to do outside of just a workout, you know, like a intense workout. But stretching in coordination with a session, I think it is universally thought that it's best done after the intense activity. And the way you do the workout, you work yourself up slowly because one of the things, you need to warm everything up, and the warm-up is important. You can injure yourself if you jump into something that's too intense too quickly, both in the connective tissue, but also in terms of muscle fiber integrity. So then, my understanding is they believe that it's best if you get the most bang for your buck in terms of the stretching, the effectiveness of the stretching, if you do it after the exercise, because those muscle fibers, you've been using them, they are used to like sliding back and forth, and it's whatever, it's just ready to be like pulled out to the proper length at that point. Yeah. I don't know. I don't know if you know Dawn Strout, but she maybe could talk about it a little bit more. Okay. So, let's see. Got some time here. So there are really three phases to a twitch. A twitch is when a muscle fiber, a muscle cell, is going to contract. We call that a twitch. All right? So there is the stimulus. That's when the nerve fiber sends the signal down. And there's a little bit of latency here, about 5 milliseconds or something like that, a few milliseconds. And what is that? Why is there this latency period? What do you think? What's happening there? Take a guess. You got your finger, you're like kind of sneaking it up there. What's up? What do you think?
It's just
Itching? Come on, somebody go for it. Yeah.
has to do with the refractory period after action.
It's a good thought. It's a really good thought. So this is when excitation starts. This is when contraction starts. What's in between? Excitation-contraction coupling, right? It takes a little bit of time for all that stuff to happen. The action potential to pass over the sarcolemma into the T-tubule to cause the release of the calcium out of the sarcoplasmic reticulum to bind to the troponin, roll the tropomyosin out of the way, form a cross bridge before we finally get here. So all that stuff that just tumbled out of my mouth happens right in this little phase. All right? So then we get contraction, and then there's this relaxation. All right? The three phases of a fiber contraction. So we're going to take this diagram, and we're going to kind of add it. We're going to build on it. The first thing here is the timing of all this depends upon, there are different muscle fiber types. You guys are probably familiar with that, right? So this is sort of like the difference between white meat and dark meat. Eye muscles are like the whitest of white meat. Very fast response time. They have very fast response time. And that should make sense, right? It should make sense. So they hit maximal tension very rapidly. Whereas you have the gastrocnemius, which are the two muscles in the back of the calf that take quite a bit longer. And then there's the soleus, which is the muscle below that in the calf, which takes a much longer time. 45 milliseconds or something like that to reach its peak. And so the speed of maximal tension production is not the only thing that's different between these. Their metabolism is also different, right? So these eye muscles are able to have, they're primarily geared towards anaerobic metabolism, very rapid anaerobic metabolism, and the soleus is geared towards primarily aerobic metabolism, aerobic production, using oxygen-dependent production of ATP. So this gets to maximal tension slower, but you can go a lot further with it. People don't do reps with their eyes going back and forth like this, but you go for a jog. Right? Maybe people do reps with their eyes in certain circumstances. I don't know. Okay. Yeah. So a couple definitions I'm going to give you here. The idea of a trapé. A trapé is a stair-step increase in tension, in twitch tension, that is dependent upon this maximal tension in trapé is dependent upon the frequency or the duration of your contraction. All right. Now, if that nerve that is coming in to tell that muscle fiber to contract is we're pushing that nerve fiber into its refractory period, into its relative refractory period, and we're speeding the frequency of this up. The tail of one of these contraction cycles starts running into, so the contraction phase starts running into the previous relaxation phase. and they start adding up. This is called wave summation. All right? You're furrowing your brow, Miles. What's up?
I'm thinking about like other things.
Okay, cool. And so say we continue that, say we continue at that pace in wave summation, we're going to reach a sort of level of maximal tension, And we call that incomplete tetanus. Tetanus being like maximal contraction of a muscle. But it's incomplete. It's an incomplete one. We can push this higher depending upon changing the frequency of stimulation from the nerve. If we really push that stimulation up to where the contraction phase of one contraction cycle is bleeding into the contraction phase of the previous, we get this sort of more smooth thing, and we reach absolute tetanus or complete tetanus. Complete tetanus, yes, sir.
I was going to ask, but you may just have said, is a complete tetanus just like a cramp?
No, a cramp. So there's a couple different reasons you can have cramps. I don't want to really get too far into that, but aspects of cramps can be a muscle in tetanus. That's right. Yeah. So sure. Yeah, a muscle that is in complete tetanus is at maximal tension. It's never going through release. It stays right up there until it runs out of ATP, right? And then there's no choice for it. It's going to have to like release. You can't get it to come out. So muscle cramps come from different reasons. There's different reasons you can get muscle cramps. Like, so you can just be in bed at night and get a Charlie horse, or you can do something and you'll get a cramp from doing it. That kind of cramp is often because of damage to calcium channels in the sarcoplasmic reticulum. Okay, so let's step up from a muscle fiber to what's called a motor unit. A motor unit. A motor unit is a group of muscle fibers that are all under the same control of a single neuron. So they're muscle fibers. They're all going to fire at the same time because one nerve controls them. Okay? And in this diagram here, we have orange nerve, blue nerve, and purple nerve. And so all of these orange fibers are going to contract when orange nerve fires. All of the purple ones are going to contract when purple nerve fires. And likewise with blue. So each of these, there's 3 motor units here. Then if we look at tension in the whole muscle as a function of time, motor unit 1 contracts and then relaxes. But motor unit 2 comes in and pushes tension in the whole muscle up, even though tension in motor unit 1 summed across those 5, 6 muscle fibers is going down. The whole tension is going up because whatever tension that's here, which is going down, is being added to the tension here that's going up. All right? And so each of these motor units add together to bring the whole tension in a muscle to whatever level it needs to sit at. All right? So the more tension that you produce in a muscle, consequently, the more motor units that are going to be firing simultaneously. Does that make sense? All right, so that was the last of my part about like the mechanics of muscles. Now let's pay for it. What do you say? Let's be responsible citizens here. All of it depends on ATP. And ATP is, the amount of ATP depends on how much oxygen and organic energy sources like glucose and fatty acids that you have. So what are we doing when we're moving muscles? We're essentially just doing this. It's combustion. It's combustion in an incredibly controlled manner. That's all that it is. That's all our bodies are. We're just bonfires. When you're burning something, you have a carbon source, oxygen comes in and oxidizes the carbohydrates, the cellulose, in the carbon source and gives us a highly oxidized carbon output. All right? That's it, and it releases energy. That's all this. That's exactly what's going on here in our muscles. So we take a carbon source in, we eat food, right? It can be fat or sugars or whatever. We breathe oxygen in, and then all that carbon that we ate as food, we breathe out as carbon dioxide. Like the actual atoms of carbon that we ingest get breathed out as oxidized carbon. It's pretty amazing. You guys, maybe you already knew this or something, but I remember when I first learned that, I was like, whoa. Oh, so cool. All right. So we're gonna talk about how that works. Different ways the body has of managing ATP supplies. And the way we're going to walk through each of these steps, I'm going to walk through them based upon time scale, the time scale of the need. All right, the time scale of the need. Do I need it like right now, just for a second? Or do I need it, for the next 6 seconds? Or do I need to really run because that werewolf's after me? Or do I need to go for a leisurely jog for the next hour and a half? Each of those has different things that are going to supply them. So we're going to start on the short time scale. And we're going to start with creatine phosphokinase. Creatine, you maybe have heard of creatine. Are there any weightlifters? Anyone ever, a little bit of you? Yeah, okay, you, maybe a couple of you guys. So a lot of weightlifters like to take creatine. They take creatine supplements. And this is why. So what happens is this protein here, creatine phosphokinase, catalyzes the addition of, so you take an ATP and you're going to take one of the phosphates off that ATP and make ADP and create creatine phosphate. So creatine phosphokinase catalyzes this reaction where creatine gets phosphorylated. Here it is. It gets phosphorylated. You may think to yourself, what the hell? That burned an ATP. I didn't want to get rid of an ATP. I wanted more ATP. Well, it turns out that this happens when you guys are chilling out right here. No problem. Don't need the ATP. You're sticking it in the bank. This is the bank. This is the bank right there. And that's not just the bank. That's like your wallet. You're sticking some 20s in your wallet so you can get them out real quick, right? So you'll notice this is a two-way reaction. It goes forwards and backwards. When the ATP supply in the ATP supply in the cell goes down, this reaction goes backwards. All right, this reaction can go backwards, and we can get that phosphate out of the bank super quickly and transfer it from creatine phosphate back onto ADP and make ATP. So it's a way of it's basically creatine acts as a phosphate reserve, if that makes sense. Does that make sense? It's pretty easy.
I've actually taken creatine before.
Yeah.
I got jacked.
Yeah.
This summer.
Yeah. Yeah. It's, creatine, the way it works in the metabolism and the things that it interacts with is actually more sophisticated than what I have I'm showing you here. But this is the basic concept. All right. The next thing is myokinase. So myokinase is, this is not a long-term strategy. This is not a long-term strategy, but I need some ATP really quick, and just for a little bit here, all right? And I've been working. I've been doing something. My ATP is down. My ATP is kind of low. I have a lot of ADP around, and I just need a little bit more, just a little bit. And so what I'm going to do is use myokinase, also called adenylate kinase, and it is going to transfer a phosphate off of ADP onto another ADP and make one ATP and an AMP. All right? Because neither of these things are useful to me. At least one of these things is useful to me. All right? You see what that's doing? So that's myokinase. Next strategy, and I probably should go to anaerobic first, but I'm doing aerobic. This would be the longer timeline. But well, it's good to talk about this. I imagine many of you have had this in the classroom, but many of you probably have not, so I'm going to go through this, and bear with me. This is like a pretty high overview of central metabolism, right? This is a pretty bird's eye view of central metabolism, and I'm not going to ask for much more detail. Is this like making anybody anxious seeing this kind of stuff on the board? Everyone cool? All right. So here's your blood, and here's lunch or breakfast, whatever, your blood sugar, and here's some more of it. All right. So we got fatty acids and sugars, and we're breathing, thankfully. So glucose comes in, and you are just chilling out. You're not doing anything. You're just sitting there listening to me talk. So we're going to put this stuff in the bank. The glucose, that's high-quality energy, right? We're going to store that as some glycogen. Meanwhile, these fatty acids that are coming in, you're burning fat, just sitting there. That comes in and goes through what's called beta oxidation, gives us some acetyl-CoA. Those are details you don't need to worry about. That comes into the mitochondria and along with the oxygen. So all oxidative energy production, energy that uses oxygen or aerobic metabolism happens in the mitochondria. All right? Anaerobic stuff happens elsewhere in the cell. But if it's aerobic, it's happening in the mitochondria with oxygen. So this oxygen is going to take that fatty acid and use the energy of it. It's going to go through the citric acid cycle and the electron transport chain, all that stuff that maybe some of you have seen before, which we're not going to go through in detail. But ADP gets converted to ATP with using this energy. So we're breaking carbon-carbon bonds, using that energy to make ATP. And then some of that ATP, we're using it to form glycogen. Some of that ATP we're putting in the bank as creatine phosphate. All right? So right now, when the economy is good, you pay off your debts. You put money in the bank. When you're making money, you put it away. You invest it, right? You don't run up the deficit. And so that's what we're doing here. We're putting glucose in the bank. We're putting phosphate in creatine phosphate. And what's coming out? We're getting some CO2 out. So this carbon dioxide, when you're just sitting there, that carbon dioxide you're breathing out came from fat. All right? That came from fat. Now, we're going to get up and we're going to move around, or at least I am. I'm moving around, and I am going to still keep burning the fat, but I'm going to start moving things in the other direction a little bit. I need to utilize my reserves. So I mobilize the glycogen into glucose. This goes through glycolysis, central metabolism, gets broken down, gets cut in half into pyruvate, pyruvic acid, which is, so glucose is 6 carbons. Pyruvate is 3 carbons. So for every glucose, you get 2 pyruvate out. Gets cut in half. And then pyruvate goes into the mitochondria and helps drive this process. So glucose getting broken down to pyruvate, this is anaerobic metabolism. Glycolysis is anaerobic. You'll notice there's no oxygen over here. But this only gives us 2 ATP. We broke that carbon-carbon bond to go from a 6 thing to a 3 thing. We got 2 out of that. But the pyruvate can go in here, and this mitochondria is magical. It gives us 34 ATP, just coming out of nowhere. We haven't touched our creatine phosphate because we're not in like ATP emergency or anything like that. Okay, so now let's do it. Anaerobic time. So when you get to an anaerobic phase, your cardiovascular system cannot deliver, you're past VO2 max, right? The demands of your cells, your muscles, for oxygen are past what's called VO2 max, and that's the maximal volume of oxygen that your body can deliver, your cardiovascular system can deliver. Once you go past your VO2 max, we get into anaerobic turf, and we're still breaking the glycogen down to glucose, and then breaking that down to pyruvic acid, but the mitochondria can't pick it up because we don't have enough oxygen in the cells. We don't have enough oxygen. We're not able to breathe enough quickly enough. And so pyruvate gets converted to lactate, lactic acid gets exported to the cell, exported out of the cell. It goes to the liver. Let the liver deal with it. But we're only getting 2. Back the last slide, you'll notice for every glucose molecule, we were getting 2 plus 34, 36 ATP for every 1 glucose. That's not bad. But here, we're only getting 2 ATP for every glucose. Much less efficient way of burning energy. Much less efficient. And so, because of that, we're gonna dip into our emergency reserves and mobilize some of the creatine phosphate. Okay? Did everybody follow that story? Metabolism? So here's a sort of survey or a summary of these different ways. So there's always just a little bit of ATP in the cell, right? So that its initial quantity is about 3 millimole. That can give you about 10 twitches in a muscle. There's enough ATP in a cell to give you 2 seconds of muscular contraction. After 2 seconds, you're out of ATP and you got to do some other thing. Creatine phosphate will give you about 15 seconds. So that's pretty good. That's 8 times the amount of cash on hand that you have in terms of phosphate that's been stored in the bank. And then just anaerobic glycogen reserves. So if you're just anaerobically burning glycogen in a cell, you got about 2 minutes. About 2 minutes of anaerobic glycogen in your cell. But if you do it aerobically using the mitochondria, you can get about 40 minutes. So they say that aerobic activity, if you want to completely, if you're thinking about your liver and your muscle and the basic health, the optimal, in terms of metabolic health, not necessarily other concerns, but like cardiovascular concerns, but metabolic health, the optimal length of an activity is about 40 minutes of aerobic activity. That's just what your cells have. Okay. All right. So this is, again, just the same recap, just kind of putting the time sequence of it in different terms for you. So here we go. We start out, and we're using some oxygen. So don't forget that we have myoglobin. I talked about myoglobin as being related to hemoglobin. It's a little bit of oxygen that we've stuck in the bank, particularly in muscles like the soleus. Particularly in muscles like the soleus or the gastrocnemius, these slow-acting, aerobically tuned muscles that really want oxygen to work. All right? That's why they're red, is because they have a bunch of myoglobin in them. this red pigment that's holding on to extra oxygen. There's the phosphagen system. We talked about creatine phosphokinase, adenylate kinase. Those were the two. So adenylate kinase is where you're transferring one phosphate from ADP to another ADP and getting an ATP and an AMP out of it. And then creatine phosphate is putting a phosphate in the bank with creatine. And then there's this glycogen lactic acid. So anaerobic fermentation or central metabolism, glycolysis, giving us 2 ATPs per glucose. And then there's aerobic respiration. And this is where we're going to get 34 ATP out of the mitochondria with some oxygen. Okay. Last concept here. We're getting pretty close. Oxygen debt. So when you start running, right? When you start running, you don't start breathing hard right away, right? Because the CO2 sensors, the chemoreceptors in the carotid bulb in your body, they haven't really picked up on the fact that the oxygen demands have gone up. So it takes a little bit for you to start breathing heavy and meet these oxygen demands right away, right? But eventually you get to this level where the cardiovascular system and the respiratory system are like, oh, okay, and they're starting to deliver that oxygen. You reach your VO2 max or whatever, and then you stop. And when you stop, your heart's still pumping, isn't it? And you're still breathing. It's because you're paying back that oxygen debt. This is I know, again, man, I'm really feeling this shutdown. Really am feeling it. I'm very good at putting that kind of thing aside, but this is what deficit spending and debt. This is the relationship between deficit and debt. And it's good for you guys to understand that because we have an enormous deficit right now. The deficit for the first time ever this year went over $1 trillion in the United States. That money, that deficit spending, has got to get paid as debt sometime. And it's not going to be paid by the old people that are making those decisions right now. That sounds really bad. I don't know. Anyways, oxygen debt. There's this deficit of oxygen that needs to get paid, which is why you breathe heavy after an aerobic activity. Okay. The Cori cycle. I don't really care about the Cori cycle. This is how we turn over lactic acid. Let's see. I got 5 minutes. Let's see if do you have any questions? Maybe I can look and find the most interesting thing that's left for me to say. This is going into the fast glycolytic versus the slow oxidative fibers, the white versus red, dark muscle fibers. Yeah, let me find. Oh, okay, I can talk about that. A little bit of pathology, why not? Amyotrophic lateral sclerosis, ALS. It's a muscle atrophy. So what happens here is, has anybody known anybody that's had ALS before? I actually decided to go to graduate school because a friend of mine, Vince Harris, he was 32 years old. He was, I don't know, he was quite a bit older than me, but I was in my 20s. He was diagnosed with ALS, and then I spent 18 months with him in hospice helping him die. I watched that, and I was like, oh my God, I got to go to graduate school. I don't know. But, and then when I finished graduate school, just as I was finishing, my father, that's him right there, he was diagnosed with ALS and passed away 5 years ago from it. It's a pretty horrifying disease, actually, if you've ever experienced it, seen anybody with it. What happens here is it seems like it would be a muscular disorder, but it's actually not. It's a denervation problem. So it's actually a neurologic disorder where the motor nerves, the muscle nerves, die. And when you de-innervate a muscle fiber, when it's not being stimulated, it itself begins to dwindle and reduce, and eventually you lose muscle control. So when you look at these cross sections of muscle, when you look at biopsies from this, it's pretty interesting. There's this patterning throughout a transect of the muscle where you can see, because it doesn't happen uniformly through all the motor units. Certain motor units will get hit first. So as you look at like a time sequence of muscle contractions, you can actually identify, oh wow, that's a motor unit. Now the additional motor units get added with time. They have shown that creatine supplement actually increases life expectancy in people with ALS. I saw that firsthand. Anyways, yeah. Are there any questions?